Welcome back to Giving Tuesday Live here at the Boyce Thompson Institute. I am Keith Hannon, and we are now joined by two of our esteemed researchers from Michelle Heck's lab, which is a big focus on citrus greening disease. We're going to talk about that in a minute. First, I want to jump back to, I made a reference to the fact that BTI was started and founded by a $10 million contribution from William Boyce Thompson in 1924, and I made a reference to how much money is that today. I had the number, couldn't remember it then. Now we have it. That would be $147 million today that was used to found BTI. So there's your fact of the day in terms of adjustment for inflation. So just to make sure we're all set there. So this is Annie and John. Thanks for coming on the broadcast today and talking to us about your research. And just maybe quickly tell us, how long you've been at BTI, kind of how you got here.
Sure. Okay. Well, hi, I'm Annie. So I am a PhD candidate in Michelle Heck's lab here at BTI. So this is my 5th year here doing science at BTI. I'm from Ohio originally. I did my undergraduate, my bachelor's at University of California, Berkeley. I studied genetics and plant biology, and so I really fell in love with plant pathogens. And when I was interviewing for graduate school, I met Michelle, and her research is so exciting, and she's such an inspirational scientist that I just had to do work in her lab. So that's kind of how I got here, and I know we're going to talk your ear off about our research. So
yeah, we're going to get there. John.
Well, I've worked in Michelle's lab here at BTI for around 5 years, and before that, I also did my PhD at BTI in George Jander's lab. I had worked with aphids in the Jander lab, and so it was an easy transition to work with psyllids in Michelle's lab. They're a similar insect in a lot of ways, and Michelle's lab also works with aphids as part of the research program that involves viruses, which is an addition to the citrus greening work in Michelle's lab.
So citrus greening, that's the big topic for this interview, and kind of what you two tackle kind of on a daily basis here. Does either one of you kind of want to give us a very kind of high level view of what citrus greening disease is, kind of where it's happening, and kind of what are the big impacts of what's going on with this disease?
Yeah. I think we'd love to. Do you want to start it off, John?
Sure. I guess the big impact on this disease is the livelihood of the citrus growers in America. Until 20 years ago, the insect that we studied, the Asian citrus psyllid, had never been found in America. And it's an invasive insect that first came to Florida 20 years ago in 1998. And in those last 20 years, the Florida citrus industry has experienced severe contraction and losses due to this bacterial disease that this insect spreads. All citrus varieties are susceptible to the disease, and at this point there really aren't any resistant citrus trees out there. So there's been a lot of challenges, first for growers in Florida, but the disease has spread across America. There's a lot of people don't know, but there is significant citrus production in Texas, where the disease has spread significantly, also Arizona, and now in California, where the disease has been more of a problem in people's backyard citrus trees, something we don't have a problem with here in Ithaca, but if we lived in California, certainly all of us would probably have a lemon tree or two in our backyard. And so it's been more of an issue with the residential trees in California, but the commercial growers are concerned. And as part of our work in this lab, Annie and I have both had a lot of contact with the citrus growers in California, and so we can really see the impact that this is making, not just on the industry, but on the individual farmers that make up this industry. And some of these California farmers that I've gotten to know, they're fifth generation citrus growers. Their ancestors have been growing citrus since the 1800s in California.
Wow.
So this is really a lot of what motivates us to find a solution to help alleviate the problem of this disease.
Yeah. And then I guess just to add to that, what you might see in a tree in a field when your field becomes infected with this bacterial pathogen is at first your leaves on the tree might have this sort of yellow pattern, which we call a blotchy mottle. And then eventually the fruit on the tree starts to taste salty and bitter, and it doesn't ripen properly. So that's actually why the disease is called citrus greening. And then actually there's something called fruit drop, where the fruit will just drop off the trees. And you can imagine that you can't harvest that fruit, so you're losing a lot economically already. And then eventually this bacterial pathogen actually kills the tree. It's completely lethal, and there's no treatment that we know to prevent tree death. So even as consumers, we're seeing effects of this disease because you might have noticed in the grocery stores, but orange prices have risen about 150% since the disease has been an epidemic in Florida. And then you're also seeing more juice blends, such as orange peach and orange mango. And that's because since the fruits taste a little bit more bitter and less sweet, some producers have to blend them to make them taste better to salvage some kind of economic profit.
And that's what I find is when I share this research with the general public, shall we call them, that is the thing that people actually notice. Some people might notice that the cost of orange juice has gone up, but people definitely, when you say it to them, they realize, oh yeah, wow, there are a tremendous amount of juice blends now that I'm seeing in the supermarket, and then it kind of dawns on them that why that is, because to your point, I mean, you know probably the numbers better than I do, but some of my research has said we're talking about $5 billion in damages over the lifetime of this disease so far, maybe more.
And
thousands of jobs.
Yeah.
So this is a huge, not only if you're an orange juice lover, is this an inconvenience for you, but people make their livelihood off these farms, and the country depends on this as a major economic export.
Absolutely.
Yeah, there's a lot of value that comes out of this industry, and it's beyond just the buying and selling citrus, but it can be the linchpin of an entire regional economy. I think one of the significant aspects of citrus production in America that probably a lot of people don't know is that Florida really turns most of their oranges into juice, whereas California, most of those oranges are sold as fresh fruit. And so one of the real challenges in Florida is that you have these juice production facilities that are built to have a much higher capacity of fruit be processed there that is currently being harvested, and that puts a lot of pressure on the whole industry, and certainly that's something that people are working for, is to make sure that these big juice processing plants will stay online, and there is progress that's being made. There are developments moving forward for trees that may not be resistant to the disease, but that can maintain a high level of production in spite of the presence of the bacteria. So while the research that we do here in the Heck Lab really has as its main focus the insect, and how does this bacteria move through this insect during the transmission, that's really what our major expertise is here, although we have many different collaborative projects. But there are whole other teams of researchers that are working to develop resistant citrus varieties, and this there's a lot of resources being put towards this problem, and we're hopeful now that the work that we do can help turn the tide, and that there's something to be hopeful about.
Yeah, I think one reason I feel this particular topic in this research is such an important story to tell is that it illustrates for everyone how it's not just about being a juice drinker and is that an inconvenience, or am I going to have lemons for my iced teas, or my cocktails, whatever it might be, but it shows the impact that crops and agriculture have on, as John said, regions, states, countries, when this is something that you've always relied upon to sustain a way of life, not only feed people, but as a business. I mean, that has huge ripple effects when you lose that. And if anyone goes on Google and searches images for citrus greening disease, you will see these really unfortunate images of just abandoned orange groves in Florida. So it's a big problem, and you're doing really important research to try to turn the fortunes here. So we're going to segue there we're going to look at a brief video about some of your new research, and then we'll come back and we'll talk about what we just saw in the video a little bit. Sound
Okay.
good?
Sounds great.
All right, Mike Carroll, who's behind the scenes today, in case you were wondering, he's the one making all this stuff happen. So he's gonna send us a video right now, real quick.
Citrus greening disease is a fatal bacterial disease which affects all commercial citrus varieties. In the last 15 years, this has resulted in the loss of approximately 60% of the citrus production in Florida.
It's caused by a bacteria, which is abbreviated as CLAS, which can be transmitted from plant to plant by an insect called the Asian citrus psyllid. So this disease makes all types of citrus fruit taste bitter, salty, and unmarketable.
The bacterium has to spread through the insect body. One of the main interests of the Heck Lab is to understand the role of the insect immune response to that bacterium.
CLAS causes an infection in the psyllid as well as in the plant. So we looked at how the hemolymph can be affected by CLAS.
This involved extracting the hemolymph from these very tiny insects and doing mass spectrometry analysis.
We found that there are very specific changes in fat metabolism which indicate that the insects are experiencing a higher level of hunger. We also found females have more of this specific immune protein called vitellogenin.
One of the critical strategies is to render the insect unable to transmit the disease without harming beneficial insects and the environment.
And we hope that understanding the psyllid immune system and how male and female insects respond differently to CLAS can help us stop these insects from transmitting this devastating bacteria.
Okay, so great video. Thank you both for making yourselves available for yet another on-camera appearance. So we'll talk about the research you did that you published this year that video was kind of based on. But let's give everyone just a brief kind of play-by-play of how this disease is transmitted. So take us to a field or a grove where this is happening, and how does this all begin?
Right. Okay, well, I'd love to take that one.
Take it, Amy.
Okay. So probably you saw in the video a picture of this insect, right? And we call that, it's called the vector for this pathogen, which means that it can spread the pathogen, the bacteria, from tree to tree. So basically what happens in a field is this insect loves to feed on citrus trees of any kind, and it has mouth parts. It has sort of a piercing, sucking mouth part that it uses like a straw. And so it lands on a citrus tree and it pierces into the tree to find the phloem where all the sugar and sap is. And then it sucks up the sap using its straw-like mouth. And so as it sucks up the sap, it also sucks up the bacteria. And so the bacteria can move in through the mouth part, the straw, into the gut of the insect. And then we know, because of work that we've done, we know that it moves into the cells of the insect's gut and then out of the gut into the blood. And there it can circulate to reach the salivary glands of the insect, and then it can be spit into the next tree. And that's basically how transmission of this bacteria happens. And the fact that this insect is really small and it can fly and it can be blown from field to field by winds or hurricanes actually enabled this disease to reach such a horrible epidemic stage in Florida. So that's why we really focused on this insect and the transmission of the pathogen as a target for how to prevent spread of the disease.
John, do you want to add anything to that, or is that pretty good?
Well, I think Andy covered it. I think some relevant things to know about this insect is that it does have a relatively fast generation time, and there's just a lot of ways that the insect can hide within the canopy of the tree from insecticide sprays. There's just many elements of trying to manage this insect that are really challenging. And another aspect of these insects is that they secrete honeydew when they feed. That's basically like the sugary byproduct of them eating all of that phloem sap. And so when that gets all over the plant, there can be secondary fungal infections that arise. So there's a lot of challenges that are presented by these insects. Now, at this point, there's been a reliance on insecticide usage to kill these insects, but that's really not proven effective, nor has it generally proven effective in strategies to alleviate problems with insect pests. It's really hard to eradicate a whole insect from your production area. So that's why Annie did a great job of explaining this transmission cycle. And the real important fact from that is it's a complex process within the insect to successfully take this bacteria in and spread it. And only this one insect in North America can do that. And so we're trying to learn about the details of how the bacteria and the insect interact within the insect with the hopes of finding a new control strategy that doesn't rely on killing the insect, but on blocking the ability of the insect to transmit the pathogen.
Great. And so the video we just watched touched upon a publication you put out earlier this year. So, how does that research depicted in the video kind of work towards or add another step to the quest to find that solution?
Sure. Well, John did a really good job explaining of how the relationship and the interactions between the bacteria and the insect, they're extremely specific, or else every insect would have to vector every pathogen, and it would be a disaster, right? So,
We don't want that.
We don't. So what we were looking at is how the bacteria interacts, but also has an effect on various insect tissues as it moves through them. And so this time, we were able to extract the blood of the insect, which actually is called hemolymph. But this blood, this fluid, is actually the center of the insect's immune system. And so we thought this would be a really excellent target because we know the bacteria moves through the blood, and then also it's an important immune tissue. So what we did is we took tiny capillaries or needles, and we stabbed hundreds of insects to extract very small amounts of blood. And
Sounds violent, but
It was
not that violent.
But then we took that blood and then we analyzed it for looking at the changes in the protein level that were caused by the bacteria. And do you want to add anything to that?
Well, I think this is just a great opportunity to talk about the fact that one of the expertise, the technological expertise areas in Michelle Heck's lab is the use of mass spectrometry-based proteomics. And I think it would be maybe useful to just take a step back and
Sure.
explain to people what that means. I think that maybe a lot of folks are familiar with the genome and genomics and that concept, and essentially the proteome is in any cell, tissue, organism, what are all of the proteins that are expressed by the genes in that genome. So the genome is going to be the same in every cell of this insect, but the proteins that are expressed in the gut versus the salivary gland versus the hemolymph may be completely different. And so the way that we analyze the proteins in these samples, the proteins which comprise the proteome, is by using this technology called mass spectrometry. And essentially, that's just a really accurate scale that tells you what the mass of the molecules that you analyze are, and using a lot of bioinformatics tools, that mass can tell us a tremendous amount of information about what the protein is that molecule, of the molecule that's being recognized. So that's just one of the major strengths that Michelle has built in her lab is our mass spectrometry skills, which have allowed us to take these very tiny biological samples and get a huge amount of biological information out of them.
Great. So where do you see, where do you maybe want or hope that kind of the next steps of the research goes, or what are you personally maybe excited to explore further within your own areas of this research?
Yeah, well, I know we're both going to have a really different answer for that because we both have a lot of different research projects
Right.
going on. From my perspective, I think that a lot of the work that we've done and the work we just talked about included helped us understand the immune system of the insect and how it interacts with the bacteria. So what I'm really excited about is developing new inhibitors against important immune proteins from the insect that might help us develop new control strategies. So basically, what I'm hoping to work towards is using the knowledge we have about the immune system and transmission and developing alternative to insecticides, so alternative molecules that aren't as damaging. And then the other research project that I'm really excited about, I think will have a more direct impact on growers, is looking at the effect of common agricultural applications like sprays and other pesticides, and looking at how they affect the insect and how they could affect transmission. And so I'm really excited to do that work and then give that information directly to growers. And I see that as being really compatible with the work that I'm doing, which might be a little bit longer to the field, which is developing new treatment strategies.
Great.
Well, it's a great question, Keith, and I guess I'll take that opportunity to talk about a couple of really exciting areas of research that we're pursuing right now. And first of all, I think it's really notable to get back to just how complicated this transmission process is. What we've identified through the research in our lab is that from going out into citrus groves in Florida and collecting insects and bringing them home and characterizing them, we've been able to identify that there's certain strains of the Asian citrus psyllid that are not effective at transmitting the citrus greening bacterium. And so a lot of our research moving forward is looking to understand what is the basis of that genetic diversity within the insects in ability to transmit the pathogen. And if there's a way to, like I said before, it's really not a realistic approach to eradicate these insects. They're everywhere in Florida. But if there were a way to tip the balance of the population so that a majority of the insects are not able to transmit this bacterium, that's a really promising strategy that a lot of the growers that we've talked to are really excited about. I guess I'd like to take a step back to talk about a different approach to controlling this insect that's really specific to this class of insect. I talked about how psyllids and aphids have a lot in common, and other sap-sucking insects. Part of their biology that's really unique are the relationships that they established with other helpful bacteria called endosymbionts, where there's a mutualistic relationship that's evolved over millions or hundreds of millions of years between these bacteria that live within specialized organs that the insects makes just for them. And specifically, there's a bacterium called Proftella in the Asian citrus psyllid that's entirely unique in all of life. There's no bacterium that's ever been identified that's really remotely similar to this. And part of the research that we've done here has actually been in collaboration with Frank Schroeder's lab here at BTI. He also has a lot of mass spectrometry expertise in addition to all of his other chemical expertise. And so we've characterized a highly abundant molecule called a polyketide that this bacterium Proftella makes. For what purpose? Nobody knows. And this is a question that has potential implications for the treatment of this disease. If we can find some way to interfere with that specific relationship between that bacterium and that insect, then we can feel confident that that treatment won't have an impact on beneficial insects or honeybees, which is always a concern when you're trying to kill insects, is that you're going to kill insects that you'd rather not kill.
Right.
But also fundamentally, here at BTI, in addition to research to address agricultural problems, we're also doing basic research about totally novel areas of biology, and this represents, I think, the intersection of those two spheres. It's a totally fascinating and new area of biology that has many different angles that it can be researched at, but it also potentially can contribute to a solution.
Well, that's one thing I really appreciate about, what we call basic research, or as we like to also call discovery science, because, here we are talking about citrus greening, but along the way, you're kind of uncovering a lot as you go, and you're kind of finding these new things like you're talking about. And is it fair to say that you might be trying to, work towards a citrus greening solution, but you're also kind of just developing, maybe even if in the early stages, but new understanding of different processes all the time? It sounds like there's more to it than just what you're going after.
Absolutely. Yeah, I think that's exactly what we're trying to do. I think so. Citrus greening is such an important problem right now, but I think that our lab is at a really unique intersection between working towards a problem that's so important and developing solutions, and then also doing discovery science so that we might develop solutions to problems we don't even know about yet.
Exactly, yeah.
Or discover new realities about insects or pathogens or transmission. And for example, I think John hit on this too, but we work on a psyllid, which is a really important agricultural pest, but it's also really closely related to a lot of other agricultural pests. And I think some of the things that we learn about it could also be applied to other things, such as aphids and whiteflies, which are really important transmitters of pathogens, but also just pests themselves. So something I find really exciting is that we are looking at the similarities between the feeding structures of those insects, and if we can find something that's a really good inhibitor, it could be applied not just to citrus greening disease, but other pathogens too.
And John, I'm always really interested in how you are out and talking with growers and farmers. What are those conversations like? Because I imagine these aren't all, totally science-minded people necessarily, and here you're kind of coming in and you're kind of sharing your knowledge with their experience. What's that relationship been like?
Well, first of all, I can't say enough about just the basic intelligence and curiosity of all of these growers who are so eager to learn about the science that we're doing,
yeah,
and support it. So I always get fascinating questions from a perspective that I may not have thought of before. I think that talking to the growers really helps us refine our scientific objectives, and we can get invaluable input from them that we take home and we use to inform our research. I think it's also just like I said before, it really puts a human face on the work that we do, and as we talked about, we're motivated by these basic discoveries, but we're also motivated by the human side of actually being able to support these people's efforts to maintain their livelihood and maintain their production.
Mm-hmm.
And I think, at least I see a noticeable push nationally to support various farming and farms and farmers, and I think it's important to also note that there's a lot of work science is doing to support those same individuals, and so support for science is also eventually possibly support for all those people that we've talked about today in the citrus world. I also want to give Annie a chance to boast, right? Because fairly recently you were awarded a fellowship, if I remember correctly.
I was, yes.
Do you want to talk a little bit about what went into that?
Sure, I would love to.
Okay.
So the fellowship is from NIFA, which is the National Institute for Food and Agriculture, though it's through the USDA, and so it's funding the last couple years of my PhD, which I'm really happy about, and it's funding a lot of the work that I talked about, which is developing new treatments as alternatives to insecticides to inhibit transmission of citrus greening disease. So I'm really excited about that. And it's a really good opportunity. I'm not sure what else to say, but I mean, I'm really excited.
Well, I just think it's a nice acknowledgement, right?
I
mean,
love
it's
that.
just
I love
not,
to
not
boast.
everybody gets one of those, right?
That's true. Yeah, and I'm really excited. So through this fellowship, I get to go to Washington, D.C. for project meetings to meet all the other people who got the fellowship and hear about their cool work too. So I'm
Great.
hoping that'll be a good opportunity to learn and meet some other, young people. It should be fun.
Awesome. Okay, and now we've come to the non-science question. So when you're not doing all this incredibly impressive research that has immense impact, what are you doing? What are you doing with with all that abundance of free time I know you have? John, you have a family.
Well,
so but you tell us a little bit about John outside of science.
Well, my family has a farm outside of Ithaca, a small subsistence farm. We have a garden and a chicken house. Very simple, but it definitely keeps me grounded. And I was able to participate in the BTI ground cherry project there in my garden, and so that's really what I love. I love having a little wood lot where I can harvest some firewood, and the garden and the orchard, and it's a great thing about living in this part of the world is that as soon as you leave Ithaca, or even before you leave Ithaca, you're surrounded by farms, and it really makes for a nice quality of life and a good contrast to just some of the technical stuff I do on my computer a lot.
Well, I also appreciate that John is maybe one of the two, probably three people in BTI I can talk baseball with a lot. So thanks for humoring me on that.
Yeah, I love the Red Sox, and I love going to the mountains, and there's lots that I can do and to have a fulfilling life, and working at BTI definitely adds to that.
And Annie, what are we doing outside the lab?
Ooh, we're doing things, Keith.
You don't have to talk. You don't have to. This doesn't have to be a full expose.
No, I'm kidding. But I love to play the flute. So I play the flute and the piccolo. I love to play classical music, and I love to play with orchestras. And then actually recently I joined a salsa group.
Sure.
So just in my free time. But it's really fun. I do love to learn new dance types and just generally try to move around, not sit at a desk. So that's what I love to do.
All right. Well, thank you both so much for giving us so much of your time this morning and sharing your expertise and your research with everyone out there. Hopefully, through the course of the last 30 plus minutes, you've gained an appreciation for really, this main problem of citrus greening that you're tackling is and the impacts that it has, and how beneficial your work has been and will continue to be. So thanks so much for what you've done, for how you've represented BTI locally and abroad. And if you want to learn more about John and Annie's research, look up Michelle Heck's lab on our website, btiscience.org, and you'll get all the great details about how I would call it a decorated lab. There's a lot of good acknowledgments that have come your way, so we're really proud of the Heck Lab here at BTI. We're going to take a little bit of an more extended break, not too long. We'll be back at noon with our next guests, who are Paul Debbie and Erica Fischel. We're going to be talking about tech transfer and impact of some of our intellectual property here at BTI. So grab your lunch and come back for that. And we'll be back on Giving Tuesday Live here at BTI.